So today we will discuss about the thalassemia. It is very important topic. World Thalassemia Day, May 8. You'll get direct question. So basically, thalassemia is a group of inherited disorders of hemoglobin synthesis characterized by a reduced or absent one or more of the globin chains of the adult hemoglobin. As we all know, we have alpha chain, we have beta chain. So out of these two chains, one of the chain is absent or reduced. It is autosomal recessive disorder, right? It is most common hemoglobinopathy in the world. Just remember this. It is again the question, most common hemoglobinopathy in the world, right? So it is basically a mutation. Alpha thalassemia. Let me discuss about the alpha thalassemia first. Alpha thalassemia is a mutation of alpha genes. Alpha gene in chromosome 16. Alpha gene in chromosome 16. Alpha gene in chromosome 16. Right. So these are the chains, alpha chain and beta chain. Right. So first of all, we will discuss about alpha thalassemia. So what is alpha thalassemia? Alpha thalassemia, alpha genes are deleted. There is a loss of one gene or both the genes from each chromosome 16. Just remember this. There is a mutation in alpha gene, chromosome 16, right? So basically, alpha globin chain is encoded by 4 alpha genes, right? So 4 alpha genes. So it is alpha 1, alpha 2, alpha 3, alpha 4, right? So we will discuss them one by one. So alpha thalassemia. So basically alpha thalassemia, if one mutation is there in 4 genes, gives rise to 4 types of disorders, because we have 4 disorders: alpha 1, alpha 2, alpha 3 and alpha 4, right? So one mutation in these 4 genes, these will give rise to 4 types of disorders. So first of all is alpha 1 gene. So what will happen if alpha 1 gene is deleted? That will lead to alpha thalassemic carrier. Right. Alpha 2 gene are deleted. Then what will happen? It will lead to, it is called trait. Right. If alpha 3 genes is deleted, then it is called HBH disorder. So it resembles the intermedia beta thalassemia. Intermedia beta thalassemia, right? So just remember that. Then the if fourth gene is deleted, then what will happen? In case of 4 genes are deleted, then it is called hydrops fetalis. Hydrops fetalis. So in this case, hemoglobin in such patients is HB BARTS. BARTS. So it is again the question in NEET. So hydrops fetalis is due to deletion of alpha 4 alpha gene, right? So the alpha thalassemia is most common cause of non-immunogenic hydrops fetalis, right? Again, remember this, right? It is most common cause of, right? So in case of alpha thalassemia, if deficiency or absent alpha subunits, sometimes excess beta subunits, sometimes excess gamma subunits in the newborns, right? So there are various types: carriers, trait, hemoglobin H disease, hydrops fetalis, HB BARTs. So this is, as I have already discussed, that alpha, this is due to deletion of alpha 1, alpha 2, alpha 3, and alpha 4, right? So this is actually very important. Hydrops fetalis, you'll get direct question. In all of them, from all of them, you can get question. HB, H disease, trait, in case of minor alpha beta, alpha thalassemia, and hydrops fetalis, HB bar. Right? Now come to peripheral smear of alpha thalassemia. So now come to peripheral smear of alpha thalassemia. In case of peripheral smear, there are variable shapes of RBCs. Now there is poikilocytosis. What do you mean by poikilocytosis? Poikilocytosis. What is poikilocytosis? There are various shapes of RBCs, right? Like this, right? And the RBCs are very small, microcytic, hypochromic, so basically microcytic and hypochromic, and HP within the, in case of target, and has target cells. So these are target cells. This is very, very important. In this case, the HB is within the center of pellar. You can see here the pellar in the whole of the RBC. So this is the case of peripheral smear, right? Now, in case of HBH, which is due to alpha 3, there is tetramer of in case of HBH, there is tetramer of beta chains, right? Which form aggregates, and that gets deposited in RBC. And we will find fine, small, round structures in the RBC. So this RBC is called golf ball RBCs in case of HBH disease, right? So just remember this golf ball RBC in case of HBH disease, right? Now come to your beta thalassemia. So what is beta thalassemia? Beta thalassemia is again due to deficiency or absence of beta subunits. It is more commonly found in Mediterranean, Middle East, Asia, and Africa. Again, it is of three types: minor, intermediate, major. So this is due to beta thalassemia is due to decrease in beta chain in chromosome 11, right? So three types of mutation. So most common is nonsense point mutation. Most common is nonsense point mutation, right? So most common is nonsense point mutation. Just remember this again. Now it is of three types. One is due to splicing. Second is due to chain termination. And third is due to chain promoter. Now, in case of splicing, there is defect in mRNA processing. mRNA processing, right? And or there is absence of RNA, which leads to nuclear degradation, and that will lead to partial reduced beta chain, or maybe complete beta chain absence. So this is called beta plus thalassemia. This is called beta O thalassemia. If there is absence, then beta O thalassemia. Otherwise, it is beta plus thalassemia, right? So three types of mutations: splicing, chain termination, chain promoter, right? Now, in case of chain termination, there is no translation and there is no beta chain synthesis. That will again lead to beta O thalassemia. And in case of chain promoter, there is decrease in transcription, which will lead to beta plus thalassemia. So just remember, in case of splicing, both types of thalassemia are there. Both types of thalassemia are there. In case of chain termination, beta O thalassemia is there. And in case of chain promoter, there is beta plus thalassemia. Right? If there is absence of beta chain, that will lead to microcity hypochromic anemia. Why? In case of decrease in beta chain, increase in alpha chain synthesis. That means abnormal aggregates are there. That will lead to destruction of cell membrane or formation of immature cells. Or there may be destruction of precursors in bone marrow also. That will lead to decrease in number of RBCs. That will lead to microcytic hypochromic anemia. Right? So there is lesser number of RBCs that will lead to decrease in formation of RBCs that will lead to microcytic hypochromic anemia. Just remember this: microcytic hypochromic anemia is due to decrease in beta chain synthesis or absence in beta chain synthesis.